Welcome to the second episode of the BestYou channel. My name is Mimi Megaso. I'm an engineer in computer science, a designer, an entrepreneur, and among other things, I love biotech, biomedicine, and all those hacks that make us have a greater, safer, and longer life. And starting by that, I want to say that today's episode, I'm going to talk about epigenetics, because as I mentioned in my first introductory video, epigenetics play a big role in what we are discovering diseases are evolved from. So it's a factor that implicates a lot of, let's say, correlations between our systems and organs that develop diseases, and sometimes they are caused by our epigenome. So let's dive into that. And I just want to thank you for tuning in, and I hope after this episode you can find some clearance and some knowledge of what epigenetics are and how you and your lifestyle can change or can regulate your systems and whatever disease you are prone to by attacking your epigenome, and let's say it in a good way. So let's start by saying what's epigenetics, right? Epigenetics, according to data, is how cells control gene activity without changing the DNA sequence, and that means that epigenomes, or the epigenetics, that is this science, is involved in the outward part of our genome, of our chain of DNA. So it covers it, it's on top of it, it surrounds our DNA, our genome, and therefore doesn't mutate per se the inside of our DNA, but it can alter it. So it brings a switch button, and that turns on and off those genes that can then become a problem. It's like certain markers that we put on top of this line of this genetic makeup that we all have. And therefore, epigenetics is associated with our lifestyle, our generational lifestyle from our ancestors too. So we can inherit some epigenetics, not genetics per se, from our parents and the trauma they suffer, even our grandparents, and there is some research that goes along that way. And let me also say that these modifications that are attached to the DNA do not change the sequence of the DNA building blocks, and that's how it alters how we read our genes rather than how the genes are. So let's say, and a lot of people say, oh, I got this from my father, I got this from my mother, it's my genes. Sometimes it's not the genes. Of course, there can be something in our genetics, an alteration, a mutation in our genes that is being inherited by our family, and it's been in many generations, but it depends of how we live our lifestyle and how we adapt ourselves to everything around us, that we can mark or switch on and off that exact gene that can then cause us diseases, even cancers. So let's say that epigenetics not only play a big role on diseases, but also play a big role on our future generations, and that's how it's related to all these problems that we carry on generation through generation. And sometimes doctors cannot find what is the cause. Sometimes it's the life of your parents or your mothers and the life of the great-grandparents. There are studies with during the Civil War with prisoners of war, and the children of prisoners of war were they had a higher rate of death, 11% more than the children of those that were at war but not were in prison. So, and among those deaths, they were caused by different causes, hemorrhage, one of those, and it was inherited by the male offspring rather than the daughters. So then we think maybe something related with the sperm and with the eggs. The eggs are more difficult to study right now, but these markers and these alterations on how we read our genes could have affected those children, and also children in the Holocaust. Children of people from the Holocaust suffer, if they suffer from dietary intake that was difficult, they suffer from insulin resistance when they develop as adults, because the parents had starvation. Therefore, whenever caloric restriction they had, they adapt to it in a way that they survive. But the children of them had this as an insulin resistance, so, and problems with glucose per se. And same with mothers that suffer of obesity. Children of mothers suffering of obesity or any metabolic syndrome, children then develop these conditions later in life, or they are also born predisposed to being fat. And per se, a mother with stress during pregnancy, children can develop neurological and degenerative problems later in life. So it's very important to understand that epigenetics and the epigenome per se are crucial to know what we can target as to have better lives, have future lives, if we're gonna have children that are healthier, stronger, and that can live under the circumstances of a healthy environment. And talking about that also, let me mention this, because I think it's important to understand how our epigenome can be modified, right? And one of the main causes is DNA methylation. Let me read. DNA methylation involves attachment of small chemical groups called methyl groups, each consistent of one carbon atom and 3 hydrogen atoms, to DNA building blocks. When methyl groups are present on the gene is turned off or silenced, and no protein is produced from that gene. So this is the way how it switches on and off. We then read our genes, and then these genes can be altered or modified in their, not in their structure per se, but in the other part of the readability. Another common cause of let's say epigenetic modification is through histones. Histones are structural proteins in the cell nucleus. DNA wraps around histones, and histone can be modified by the addition or removal of chemical groups, again methyl groups or acetyl groups, and these chemicals influence how tightly the DNA is wrapped around the histones, and that can affect also how a gene can be turned on or off. And these errors in modification of brown genes can also be prone to modify the gene activity or inactivity, and then generate disorders. Conditions such as cancers, metabolic diseases, degenerative disorders have been found to be related to epigenetic errors. So there we have a very wide spectrum of problems, just might be caused by first our epigenetic errors or changes. Scientists continue exploring a relationship between the genome and the chemical compounds that modify them, and these modifications and errors have on gene function, protein function, and human health. Also, in between these epigenetics modifications, we have, as I said, DNA methylation, modifications to the chromatin, loss of imprinting. A lot of loss in the imprinting can cause also that genes are not correct and therefore create disorders, and non-coding RNA, even small portions of RNA. Although epigenetics changes do not alter the sequence of the DNA, they can cause mutations. And exactly, a gene is a short piece of DNA that tells the body to build specific proteins. So these proteins are specific to certain types of genes, telling to produce. So when there are alterations, then these alterations also are modified and through chromosomes. So when there is something that is an error, that produces a disorder or a disease. About half of the genes that cause familial or inherited forms of cancer are turned off by methylation. So that's very important to understand now to develop better cancer treatments through epigenetic studies. So a DNA also is this information that is stored in codes, and these codes are made of 4 chemical bases: adenine, cytosine, guanine, and thymine. And the combination of these brings a structure that then calls for information. Our human body has 3 billion bases of these. 99% have the same in every human being. So only 1% change. But although it's 3 billion is a big number, there are flowers like the Japonica, Paris japonica, that has 50 billion bases. Being such a small plant has more bases than us. So there is no correlation between the size of them and the size of the body per se that have them. We also have a genome. Our genome consists of 23 pairs of chromosomes, but we also have chromosomes in the cells of the mitochondria, and that's very important because our mitochondria generates energy for the whole body, and we will not talk about that because it's very important for repairing our body in many, and let's say all the diseases that are involved that people are suffering now. But then let me tell you also why I'm talking about epigenetics as my second episode, because I think it's crucial. And let me read this in case you still are reluctant to understand that epigenetics is a must to know how we can change our life and how can we become better on having us alert of what this is. In the next 10 years, 1 billion people are estimated to suffer from disabling consequences of metabolic disorders, making them the #1 non-communicable disease of on a global scale by 2030. Loss of risk factors such as dietary intake, lack of exercise, and other life behaviors are considered to play a role in the development of metabolic disorders. Evidence suggests that the pathogenesis involves changes on chromatin and chromatin-modifying enzymes, which can contribute then to a persistent dysregulated metabolic phenotype. And these dysregulations are then what we call diseases or disorders, right? And these non-communicable diseases are the leading cause of morbidity and mortality worldwide. Metabolic diseases constitute the greatest part of NCDs, and according to the International Diabetes Foundation, more than 693 million people will be suffering from diabetes by 2045. That's a big number. Obesity and cardiovascular diseases are rising. A group of risk factors such as high fasting blood sugar, high blood pressure, low HDL, cholesterol levels, and other defined as metabolic syndrome, which is thought to predict or increase the risk of diabetes and heart diseases. And for that, this projection says that 1 billion people worldwide will have prevalence of obesity by 2030. So we have here a whole system which is called the metabolic syndrome that we will gonna talk later. But this regulation of the body that is caused essentially is sugar. Let's point it out loud now, right now, sugar, because the consumption of sugar in rice, in flour, in many of our foods, bread, and this alteration of the insulin and then our glucose levels generate of all these metabolic syndromes. Is many of us, we are at fault on this, on how we consume and what we consume on a daily basis. Also, environmental factors affect epigenetics and metabolism, and also a predisposition in later stages of our life to have metabolic problems or diseases. Here we say not only what we eat, also societal factors such as sleep patterns, meet timing, and work shifts can cause circadian misalignment. So we have a circadian clock, we're going to talk about that later, but if we alter our natural clock, one of our clocks, then we alter our whole metabolism. Hypermethylation of various tissues, specific clock genes, lead to increased insulin resistance and impaired glucose tolerance just with one night of deprivation of sleep. So let's think these things are not just talk. It's very important to know that every time we pass 2 a.m. watching Netflix and we are dysregulating our circadian clock, therefore we are also altering our insulin resistance and therefore exploding how our glucose is gonna be in our bodies, and that's not good. That generates metabolic syndromes and therefore diabetes in a later future. Also, it has been shown that metabolites deriving from various food sources can serve as substrates for transcription factors and histone-modifying enzymes that can affect chromatin compaction, leading to more sequestered or exposed areas of the genome to down or up-regulate the gene transcription and therefore create such errors. I know that some terms are not as easy as I would like to, and it's very difficult to wrap all this in just one video, but I will just mention these very important parts of these studies so you can understand that what we do, our lifestyle affects our epigenome and therefore affects how we read our genes and how they can interact with us in a good way or in a bad way and produce us diseases. There is this part that is very important. The methyl CpG binding protein, MECP2, plays a central role in to regulate epigenetic imprinting and chromatin compaction. Mutations in many of various epigenetic regulatory enzymes have historically first been associated with cancer. Cell metabolism and cancer are tightly linked. Many cancer-associated genes can also cause metabolic diseases phenotypes without cancer symptoms. Histone, lysine, and methyl transferase, known as KMT2D, is linked with acute myeloid leukemia, lung and colon cancer. Also is associated with Kabuki syndrome, which is a multiple congenital anomaly syndrome characterized by facial and skeletal anomalies. And these mutations in MII2, which is the same name for mice, result in insulin resistance and impaired glucose tolerance in mice. Also, KMT2D mutant mice reveal features of mild non-alcoholic fatty liver disease, elevated cholesterol and triglycerides plasma levels. Another study identified KMT2D to an NAFLD, elevated cholesterol, and regulator of hepatic cardiac clock and coactivators of the circadian transcription, which has been implicated before by controlling lipid metabolism. So DNA methylation can be hereditary maintained in developing embryos too, so we can develop this and then give it to our embryos. A number of imprinted genes contribute to control of the energy in homeostasis and glucose regulates metabolism, including IGF-2, GRB10, and the LK1. Excessive IGF-2 expression can cause a loss of imprinting. Hypoglycemia occurs in 50% of cases of a syndrome called Beckwith-Whitman, and this is caused by hyperinsulinism rather than the IGF-2, but there is an underlying correlation there. Epigenetic studies have shown that parental obesity can affect methylation of IGF-2 of the fetus, correlating these changes with birth weight and metabolic syndrome later in life. Later in another video, I will talk about how these consequences are also very implied in cancer, and drugs and alcohol can also impair our readability of the genes, let's say our epigenetics. And I will close it right now. I will wrap this video because I know there has been a lot of information, but I just want to say that if we don't care about how we live our lifestyle, we deprive our body with how we eat and how we consume these toxins, and let's call sugar a toxin too, because it's part of our energy, but indiscriminating use and abuse of sugar, and let's say in many of our foods, especially from processed and refined foods, can alter our epigenetics, and then can alter how our lives are prone to metabolic syndrome, cardiovascular diseases, neurological degenerative disorders, Alzheimer, Huntington disease, and many others. So I will leave you with just a thought that please, if you wanna think whatever you have or whatever you are prone to have is your genes, maybe not, it might be how are you living your life. And therefore I'm here to tell you we can live a better life, a healthier life, and a stronger life if we are knowledgeable about the new research, and if we know that by being us every day and having changes in our lifestyle, in our diet, in our way of sleeping, in when, how, and where we do things, can alter our healthy life and can alter the life of our future generations. So I leave it by that, and thank you so much for watching. This was the second episode of the BestYou channel. Thank you for tuning in, and I see you next time. Bye bye.